The focus of this research project is urban small mammals. People's relationships with animals have changed as the boundaries between human places and wild spaces have shifted. We have reshaped and manipulated natural areas into spaces designed for human use, influencing the ecosystem. Humans have had a negative influence on the ecosystem urban small mammals originated in, due to the habitat fragmentation, development project, and destructions. Based on a study done on the effects of urban habitat fragmentation on common small mammals in 2011, animal dispersal occurs in correlation to accessibility to resources, affecting small mammal distribution and abundance. These factors not only affect the plant community within these habitat patches, but also decrease urban small mammal diversity and abundance. Habitat patches are defined from a human perspective as we are the ones creating these gaps in resources. Based on a study done on the functions of urban areas for species diversity in 2011, in these habitat fragmentations, there are two important components to make these spaces successful. These are to have vegetation and animals and to present it with the possibilities to move through urban spaces. Based on literature review, a healthy ecosystem will have enough resources for all organisms to succeed, leading to a high species richness and abundance within the community. We assessed the small mammal population at the Bronx Zoo by evaluating these factors. We tested the health of the mammal population by documenting small mammal species abundance based on proximity to the pedestrian pathway and by surveying the plant community across various sites in the Bronx Zoo. Our research question going into this project was how the human-nature interactions affect habitat quality of small mammals in a managed green space. The nested questions we proposed were, how does small mammal species diversity and abundance vary with proximity to walking paths? And how does small mammal diversity and abundance vary with the plant community? We hypothesized that the furthest distance from the pathway at each site will show the highest species abundance because of animals' preferences for remoteness. We hypothesized in a closed managed space we would see the highest small mammal species abundance at sites with the largest percentages of native vegetation because native plants provide vital resources for native wildlife.
Now I'll explain how my research team tested these factors. First, we had to choose where we'd evaluate our research question. We chose 3 different sites in the Bronx Zoo that had different characteristics from one another. They differed in proximity to the pedestrian pathway and plant community. In order to assist the small mammal species richness and abundance, we placed track tubes and camera traps at each of these sites in order to spot the mammals that existed within them. To build the track tubes, we used 2 pieces of aluminum gutter. We placed contact paper on the bottom surface at each end of the gutter, and a piece of felt-soaked ink was secured. In the center of the track tube, there was a petri dish with peanut butter and oats as a way to bait the mammals. This allowed us to capture an image of the footprints the mammals were in that area. At each site, 3 track tubes were placed at 3 different locations. One was placed to the pedestrian pathway, another was a medial distance from the pathway, and the last one was the farthest from the pathway. We wanted to see if the difference from the pedestrian pathway had an effect on the small mammal species richness or abundance. The camera traps we used to confirm which animals were present in our sites in case we could not evaluate the footprints on the contact paper. 1 camera trap was placed at each site. We assembled and left the track tube there every Tuesday, picked it up every Thursday, left a newly assembled track tube every Thursday, and picked it up every Tuesday. At each site, we surveyed the plant community. We were interested to see if this had an effect on the mammal population. To conduct the vegetation survey, we set up subplots that were 10 meters in a cardinal direction. We recorded all tree and ground cover plant species within this subplot. We worked together to identify the plant species in our subplots using iNaturalist. After we evaluated our data and compared the various factors to see if there was a relationship between the proximity to the pedestrian pathway, plant communities, and small mammal species richness and abundance.
After weeks of data collection, we saw the following results. In graph 1, we are showing relationship between small mammal species abundance and the distance between the pedestrian pathway at site 2. As you can see, the highest abundance of mammals was the medial distance of 40 meters from the pedestrian pathway. In graph 2, we found the highest abundance of mammals was also the medial point, which was 14.25 meters in site 3.
Graph 3, located in Site 7, the highest mammal abundance was seen at 5.3 meters. In a managed green space like the Bronx Zoo, we saw plant communities differ site to site. The site with the largest percentage of native species in the overstory, mid-story, seedling, and ground cover layers was in Site 7. Site 3 had more introduced species within the overstory, mid-story, seedling, and ground cover levels. There was a presence of invasive species within the ground cover. In Site 2, the ground cover includes native and invasive species and introduced species.
We hypothesized that the farthest distance from the pathway would have the highest abundance because animals would prefer remoteness as they view humans as a threat. We saw in our results that the medial distance from the pathway was in fact where the highest abundance of mammals was seen. Based on literature review, animals will spend more time near the pedestrian pathway if they are searching for food. We believe this was the case in a managed space like the Bronx Zoo. We also hypothesized that the highest small mammal species abundance would be found at the sites with the largest percentages of native vegetation. This is not what we saw in our results. Site 7 had the highest percentage of native vegetation at each forest layer, but did not have the highest abundance of species seen. Our results show that the farthest and closest distances from the pedestrian pathway had the lower abundances of small mammals. This could be due to the small mammals being attracted to areas with dense vegetation. Site 7 had the highest percentage of native plant species but had the second ranking for abundance of mammals. We saw that the highest amount of unique species at Site 7. What is not documented in our plant survey is the percentage of invasive vines that cover the site. More research is needed to understand the relationship between the urban mammals and invasive species. Using the information we gathered in our own project can help with knowing what areas to place track tubes, camera traps, and future urban planning, and more specifically, a managed closed space.